So what Hardy-Weinberg does then is presents us with an equation that we can use to figure out whether or not a population is changing. Because if we know what a population is like when it's in equilibrium, then we can figure out what it looks like when it's changing. So for all populations in equilibrium, this equation, p squared plus 2pq plus q squared equals 1, holds true. So you definitely want to write this down. Down below here are the kind of keys to what these different variables mean. So the p squared is the homozygous dominant frequency. Right, so we got to do a little bit of a reminder here of some of these terms: homozygous dominant, homozygous recessive, all of those things. Let's see, let's write in yellow, how about? So homozygous dominant would be like big D, and frequency is equal to a percent or a decimal, because really decimals and percents are kind of the same thing depending on how you write them. So basically what we're saying is p squared is the percentage of homozygous dominant individuals in the population. So if we again look at our gene pool and look at all the homozygous dominant gene combinations in the population, that would be our frequency, our percentage of those would be our frequency for p squared. Okay, so the next one, 2pq, is the heterozygous frequency. A frequency, again, meaning the percentage or a decimal. Heterozygous would be one big letter and one small letter. And then finally, q squared is the homozygous recessive frequency. Two little letters. Hopefully these are all coming back to you now. And frequency, again, meaning the percentage or the decimal. Okay, hopefully you have those written down. We'll move on. Here's an example of what we mean by gene pools and frequencies. So we have these fields of flowers here. Let's take a closer look at those. This first field here, we're going to start on the left. Okay, as you can see, we have a pretty, straightforward variation. Some of the flowers are red, some are pink, some are white. And you can see based on their genotypes, again, the genes they carry, that they are different combinations of genes to make these different phenotypes or colors. So all the reds are big C, big R with big C, big R. All of the pinks are big CR with big CW. And all of the whites are big CW with big CW. Okay, so they have a different set of genotypes depending on what their phenotype is, which makes sense. Hopefully you remember how that works. So we're going to look at the first generation down here. It says that the P frequency, so the percentage or the decimal for the C big R, that's we're taking as our dominant allele, is 0.7. How did they get that? Well, they added up all the big CR. Here's one here, there's two here, so there's two here, there's one here. There's two here, there's two, and I'm looking for a big C, big R, or even just a big CR on its own, which counts as 1. So we're adding them all up together. There's one there, there's one there, and we get, let's see what number, 2, 4, 6, 8, 10, 13 is what my count is. Let me double check. 2, 4, 6, 8, 10. Okay, so I get 13 total dominant alleles. But remember, the frequency is the percentage of the whole. So how many total alleles do we have? Well, those 13 plus the ones that I didn't count yet, because they weren't big C, big R. So we've got one here, 1, 2, 3, 4, 5, 6. So 13 plus 6 is a 19. If we take 13 divided by 19, we get 70%, or 0.7. So that's how they found this first one, the frequency of that P allele. Q allele is the same way. We're dividing by the total again. However, this time we don't have 13 big C, big W's, because that's our recessive allele. Instead we have, do you remember what we counted? It was 6, right? We circled those up. So we have 6 divided by 19, which is a total of 0.3 for our frequency. Okay, so that's how we're finding frequencies from one generation to another. Okay, the frequency of the P versus the frequency of the Q. Now we're going to move on to another generation. So here we've got a little note that tells us only 5 of the 10 plants leave offspring. Okay, these 5 plants that leave offspring are the ones that are in white. Okay, those 5 leave offspring because the environment favors those for whatever reason. Maybe they grow faster in the sun environment that's there or whatever. So those that leave offspring, we get a different mix this time. Okay, here's our next generation. If we would count up all the big CRs and all the big CWs, add them up and divide by the total again, which I'm not going to do again, but hopefully you remember how that works, we would get these frequencies instead. Okay, 50-50 now. Half of the alleles in this second generation are big C, big R, and half of them are big C, big W. Again, this time, the environment favors only 2 of the plants leaving offspring. What happens then? The 2 plants that the environment favored were completely homozygous dominant. So all of the alleles in that population are dominant. So our p-value is 100%. Right? 100% of our alleles are the dominant alleles. Our q value, or recessive alleles, is 0. Now this is 3 generations. This would be extremely fast. Evolution, natural selection would never happen this quickly. But it gives you a good idea of what exactly is happening here and how the frequency of the alleles is what you actually measure to see if that whole population is changing or evolving. Okay, let's take a look at another example here. This, you don't have to write down in your notebook, but I'm going to take you kind of through the steps and to figure out how this works. So we're going to have a population of 200 mice. So right away we've got our total. Right, here's our 200 mice right here. 98 are homozygous dominant for brown, or big B, big B. 84 are heterozygous, big B, little b, so we're going to assume they're brown as well. And 18 are homozygous recessive, little b, maybe they're white. Okay, what we need to figure out is what are the allele frequencies, number 1. So in other words, what are the P's and what are the Q's? What are the genotype frequencies in this population? So this is your P squared, I'm just going to draw it up here, P squared, Q squared, and 2PQ. Those are the genotypes. And then what are the genotypes of the next population if equilibrium is maintained? All right, so this is the type of problem that you're going to have to work through for this Hardy-Weinberg type of situation. The allele frequencies are going to be your first ones that you need to find, and in order to do that, we're going to do kind of a percentage calculation again. So we'll get to that in just a second here.